Hello, everyone, and thank you for that wonderful introduction. I'd like to thank SBIA for the opportunity to talk to you today about process validation in ICH Q7. As noted, I'm Dave Amspacher, and I'm a Drug Master File Reviewer in the Division of Lifecycle API. What I'm going to do today is give you a first-hand account of what we're looking at when we review the information you give us in the process validation section of your Drug Master File. I'll start off today with some learning objectives. First off, we're going to talk about why the FDA wants you to validate your process and why it's important. To set the table, we'll briefly touch on how you go about validating your process, and most importantly, we'll talk about what we do with your validation data and how we use it. Start off with, I want to briefly touch on the regulatory authority involving validation. This authority is drawn from the Code of Federal Regulations in Title 21, Part 211.100. This slide showing the justification is directly from the process validation guidance document, and it states that not only do we have the authority, but the obligation to evaluate the process validation that you give us, and that there is a need for a high degree of assurance that drug products meet the attributes that they are intended to have. So to define our terms, let's talk about what a process validation is. Process validation is the documented evidence that your process can produce API that meets specifications. So what do we mean by that? Product quality is derived from careful and systemic attention to selection of quality components and materials. So there's a quality in and a quality out component here. If you start out with quality material, that's going to carry through to the product. Adequate product and process design and control the process through in-process and end-process testing. What this means is that you have to say what you're going to do, and then you do what you said you were going to do, and show us evidence that your process did what you said it was going to do. For our first challenge question, we'll ask about manufacturers submitting validation data. True or false? Most drug master files contain a summary of validation data upon initial submission. The answer is true. Almost all drug master files that are submitted contain validation data. Sometimes a drug master file is submitted before the validation is completed, which is possible. A drug master file can be submitted without validation data, but we'll ask you for it if it's not part of the DMF. This review process starts with the completeness assessment. Every drug master file that is submitted is reviewed for completeness. This is not a review of the content to see if it's adequate, but to see if there is data provided in each section of the drug master file for us to review. We'll look in section 3.2, S2.5, and see if it's there. One thing to note is that the guidance states before any batch from a process is commercially distributed, a manufacturer should have gained a high degree of assurance in the performance of that manufacturing process. So you have to do the validation before any commercial distribution. This assurance should be obtained from data from pilot and or commercial scale studies. Our obvious preference is that you submit data on your commercial scale studies because, as we all know, there are significant differences between the small scale experiments and kilogram production batches. Here's our second challenge question. In ICH M4Q, section 3.2 S2.5 states that process validation studies for aseptic processing and sterilization should be included. True or false? My process is not aseptic. It does not involve sterilization, so therefore I do not need to provide a validation. This is false. The guidance states that studies for aseptic processing and sterilization should be included, not that the validation of non-sterile processes is excluded. So why do we ask you to validate your process? We want to know that it works, that the systems, facilities, and processes perform their intended functions adequately and consistently as specified. Basically, that your process does what you say it does. This is born out of clinical failures of several products, which were caused by significant content uniformity problems that were the result of poorly controlled manufacturing processes. Also in the past, batch failures were often associated with unvalidated manufacturing processes. Effective process validation contributes significantly to assuring drug quality. Quality, safety, and efficacy have to be designed into the product. Quality cannot be adequately assured merely by in-process and finished product inspection and testing. So what do we mean by that? This ties back into our previous definition of process validation in that you need to have a fundamental understanding of the process. Things like in step 2, if the temperature goes above X degrees, this impurity forms, which is why we monitor the temperature. Or impurity X in the starting material will carry through and forms impurity Y in the drug substance. In my experience, failure during production is often the result of incomplete or faulty understanding of a process's capability. In other words, what the process can and cannot do under a given set of operational circumstances. Validation in itself does not improve processes, but confirms that the processes have been properly developed and are under control. It is also beneficial to the manufacturer because the application of process validation should result in fewer product recalls. Also, each step of the process needs to be controlled, and we'll delve into that in a few slides. So how do we go about validation? The majority of the process development activities occur either in the pilot plant or in the proposed manufacturing plant. Process development programs should collect and evaluate data. Basically, you need to run the process and document the results. This includes each stage of the process from the process design stage through commercial production. You need to establish scientific evidence that your process is capable of consistently delivering quality product. So what is scientific evidence? Data. Data collected by appropriate analytical methods. Prospective validation should normally be performed for all API processes as defined in ICH Q7 12.12 and should be completed before the commercial distribution of the final product manufactured from that API. So the first step is defining the commercial manufacturing process. As we know, the process used in early development is often very different from the final commercial process. And this process is defined during this stage based on knowledge gained through development and scale-up activities. It is important to identify the process parameters that could affect the critical quality attributes of the API. This goes back to having a fundamental understanding of what is happening in your process and why. Determining the range for each critical process parameter is important so you know how far off center you can be without compromising quality. These studies are used to gain a fundamental understanding of the manufacturing process and to determine the critical process parameters or operating variables that influence process output and the range of numerical data for critical process parameters that result in acceptable process output. If the capability of a process is properly delineated, the process should consistently stay within the defined limits of its critical process parameters and product characteristics. So stage 2, qualification. This is where you run the process to see if the process is capable of reproducible commercial manufacturing. You have to run the process enough times to determine if the process gives reproducible results. Critical process parameters should be controlled and monitored during process validation studies. This is the data used to show that the process is under control. Process validation should confirm that the impurity profile for each API is within the limits specified. The design was done in the previous step. Here, we are confirming that the process does what it is designed to do. A validation report that cross-references the validation protocol should be prepared, summarizing the results obtained. This is what you send to us and what we draw our data from. After the validation, you're not done. You have to continue to monitor and maintain the process in a state of control, even as things change. Now we come to our next challenge question. We only have to do 3 validation runs. This is false. Neither the CGMP regulations nor FDA policy specifies a minimum number of batches to validate a manufacturing process. Validating a manufacturing process or a change to a process cannot be reduced to so simplistic a formula as the completion of 3 successful full-scale batches. We strongly recommend firms employ objective measures, meaning statistical metrics, wherever feasible and meaningful to achieve adequate assurances. The number of samples should be adequate to provide sufficient statistical confidence of quality both within a batch and between batches. The confidence level selected can be based on risk analysis as it relates to the particular attribute under examination. In theory, the number of process runs carried out and observations made should be sufficient to allow the normal extent of variation and trends to be established and to provide sufficient data for evaluation. In practice, it might take some considerable time to accumulate this data. Some considerations should be exercised when selecting the process validation strategy. Among these should be the use of different lots of raw material and major excipients, batches produced on different shifts, the use of different equipment and different facilities, dedicated for commercial production, and the operating range of critical processes. So how do we use your validation data? Aim of pharmaceutical development is to design a quality product and its manufacturing process to consistently deliver the intended performance of that product. This relates back to what we said before, in that you say what you're going to do and you're doing what you said. So how do we, the FDA, know that you're doing what you said you were going to do? Aspects of drug substances and manufacturing processes that are critical to product quality should be determined and control strategies justified. This is related to the phrase control of the process. You have to run experiments, and you have to know what is going to happen if you deviate from your process, and you provide that to us as a justification. The temperature of the recrystallization of X goes above Y, impurity Z will be formed, so we have a test for that in this intermediate. This is the information and knowledge gained from pharmaceutical development studies and manufacturing experience, providing scientific understanding to support the establishment of the design space, specifications, and manufacturing control. Quality cannot be tested in products. Quality should be built in by design. You do not know what is going to happen if your process goes outside the parameters. You cannot test for it because you do not know what the problem is. You don't know what you don't know. Everything starts in section 2.2 with the reaction. We prefer a table of starting material and reagent input weights that also includes molecular weight, moles, and molar equivalents, output weight of product, and percent yield for each stage of the process. We start with what you're adding, how many moles and equivalents, and how much product you get out, and what the yield is based on the amount of starting material you're putting in. In this example, you can see there is an excess of acetic anhydride added. Because we have a reagent being used in excess, I'm going to ask myself, where does this extra reagent go? Also, since the yield is 75%, I'm going to ask myself, where is the 25% of the starting material that does not react? Also, I'm going to ask myself about impurities from any overreaction. Here are some theoretical examples of our thought processes and how we look at the validation data given to us. Opportunities exist to develop more flexible regulatory approaches. To realize this flexibility, the holder should demonstrate an enhanced knowledge of product performance over a range of material attributes, manufacturing processes, options, and process parameters. The first hypothetical example gives us very little flexibility because there's minimal control shown at each stage of the process. We start out with the starting material that has an impurity in it, which is then used in step 1. Impurity 2 may or may not be formed in this reaction, but we really don't know because it was not discussed. Impurity 3 is formed, but the only control it as an in-process test. The intermediate is formed is not isolated, so at this point we do not know where impurity 1, 2, or 3 is. The unisolated intermediate is then used in step 2, where impurity 4 may or may not be formed, but it is not discussed, so I really don't know. Impurity 5 forms, but it's only controlled as an in-process test, so at this point we do not know where impurity 1, 2, 3, 4, or 5 is. This unisolated intermediate is then used in step 3, where impurity 6 may or may not form. We don't know because it's not discussed. So now at the end in the drug substance, we're concerned about impurities 1, 2, 3, 4, 5, and 6. So this is a situation that we refer to as deferring all the testing of impurities until the drug substance. So this is why we ask for information like traceability, analytical data of starting materials, intermediate results, in-process testing results, and final drug substance results in tabular format. In this situation above, we have a starting material with impurity 1 in it, which is used in step 1, where impurity 2 may or may not form, and impurity 3, which we know is present. In this example, the firm has shown us that impurities 1 and 2 are purged into the water layer of an extraction, and they have isolated the intermediate and controlled impurities 1, 2, and 3 as specified impurities. Since there's testing of an isolated intermediate, the firm could demonstrate that their method for intermediate 1 can detect impurities 1 and 2, which could then be controlled as unspecified impurities. Now, after this point, we are not concerned with impurities 1 and 2 because we have demonstrated control. This isolated intermediate with impurity 3 is used in step 2, where impurity 4 may or may not form, and impurity 5, which we know is present. Impurity 3 and 4 are shown to be purged in the mother liquor in the isolation of intermediate 2, which controls impurities 3, 4, and 5 as specified impurities. Since there's testing of an isolated intermediate, the firm could demonstrate that their method for intermediate 2 can detect impurities 3 and 4, which could then be controlled as unspecified impurities. Now, after this point, we are not concerned with impurities 3 and 4 because they have demonstrated control. This isolated intermediate with impurity 5 in it is used in step 3, where impurity 6 may or may not form. Since we've demonstrated control of each impurity as we've gone through the process, we are only concerned with impurities 5 and 6 in the drug substance. In the release testing for the drug substance, the firm could control impurities 5 or 6 as specified impurities, or the firm could demonstrate that their drug substance related substances method can detect impurities 5 and 6, which could then be controlled as unspecified impurities. Good quality detailed validation data gives us the ability to follow the fate and purge of impurities. We want to know what impurities are possible and how they are purged and how you know that they are purged. In addition to sampling requirements, the OGMP regulations also provide norms for establishing in-process specifications as an aspect of process validation. In-process materials should be controlled to assure that the final drug product will meet its quality requirements. In-process specifications shall be derived from previous acceptable process average and process variability estimates where possible, and determined by the application of suitable statistical procedures where appropriate. The data from your development studies and validation should be used to set the limits in your specifications. One example of suitable statistical procedures that would apply here would be 3 sigma. Also, the limits should be consistent with drug product final specifications. Now on this slide, I have a few examples of common situations. The limits for the tests in the intermediate specifications need to be appropriate for the levels of the observed data. For hypothetical intermediate 3, we have a limit of 10% for impurity 1, but we only see 0.2 to 0.3% of this impurity. We understand that you want to have as much flexibility in your process as possible, but in this situation, we would ask ourselves, well, what happens if there's a batch with 9.5% of impurity 1? Will the process be able to purge that much of the impurity? We really don't know. On the flip side, we do not want to box you in and create a batch failure because of limits that are too tight, like in impurity 2. One way you can justify a limit in your process is to provide a spike and purge study. This demonstrates that your process can adequately purge a particular impurity. As shown in impurity 3, this limit indicates that you know it is there, and how much you should expect, and you will know if something goes wrong. In impurity 4, we have a similar situation as impurity 1. Because there's no example of this impurity being purged, we do not know if the system has the ability to purge this amount of the impurity. We would be less concerned with impurity 5 because the limit is low and the process should be able to purge that amount of an impurity. But if it does not, the limit is low enough to catch any problems. Impurity 6 is a situation where we see wild swings in results. This indicates a lack of understanding and control of the process. Analytical results of all batches of the drug substance should be provided in the application. This includes the stability batches and commercial scale batches. If an impurity is found above the identification threshold in any batch manufactured, it should be identified. Any degradation products observed above the identification threshold should be identified. Your drug substance specification should include a list of impurities. These impurities should be the impurities found in batches manufactured by the proposed commercial process. Individual impurities with specific acceptance criteria included in the specifications for new drug substances are referred to as specified impurities. Specified impurities can be identified or unidentified. Also, a rationale for the inclusion or exclusion of impurities in the specification should be presented and include a discussion of the impurity profiles observed in batches manufactured by the proposed commercial process. And we'll give you a little bit more detail on organic impurities. You should summarize the actual and potential impurities most likely to arise during the synthesis, purification, and storage of the new drug substance. These impurities should be based on sound scientific appraisal of the chemical reactions involved or impurities associated with raw materials. These impurities should be limited to those that might reasonably be expected based on your specific knowledge of the chemical reactions and conditions involved. In summary, process validation is the documented evidence that a process can produce an intermediate or API meeting its predetermined specifications. Effective process validation contributes significantly to assuring drug quality. There are opportunities to develop more flexible regulatory approaches. To realize this flexibility, you have to demonstrate an enhanced knowledge of product performance over a range of material attributes, manufacturing process options, and process parameters. Thank you all very much for your time and your attention. And if you have any questions, I'll be available in the panel that's coming up.